Hello, this is Yashveen. In this video, I'll be solving paper 2, variant 1, May-June 2019 9700 biology. Starting with question number 1a. Antibody molecules are proteins that show primary structure, secondary structure, and tertiary structure, and quaternary structures. Figure 1.1 shows a ribbon diagram of an antibody molecule. So antibody molecules are globular proteins with a three-dimensional structure. Describe how figure 1.1 shows the secondary structure and the tertiary structure of the antibody molecule. Secondary would be made up of alpha helix and beta helix, but since this diagram does not show any particularly tighter coils of polypeptides and only looser ones, so I'll be mentioning only those. In the tertiary structure, it's represented by the three-dimensional shape of the polypeptide by folding the already folded polypeptide. And this would be formed by forming bonds like hydrogen bonds, ionic bonds, weak hydrophobic interactions, or disulfide bonds. B figure 1.2 is a transmission electron micrograph of a hybridoma cell. So a cell that is formed by fusion of plasma cells along with cancer cells. You'll be able to see that X is a rough endoplasmic reticulum, so it's got ribosomes attached to it. Now, ribosomes on the rough endoplasmic reticulum are the site of protein synthesis or the polypeptide synthesis, and the rough endoplasmic reticulum itself is responsible for modification of those polypeptides. Y, when you look at it, is a relatively larger organelle which has got projections which could be cristae of mitochondria, so I'd assume that's what the structure represents over here, and it is responsible for providing energy in the form of ATP molecules for protein synthesis. 2. The hybridoma method for the production of monoclonal antibodies involves a number of stages. One of these stages is the formation of hybridoma cells. Outline the stage in which hybridoma cells are produced. So these cells are produced when plasma cells fuse with cancer cells. The plasma cells are the ones that have been collected from the spleen of a small animal like a mouse. When it's injected with a particular antigen against which you want to synthesize particular antibodies, and so those specific plasma cells would be present in the spleen of this animal where you'd be able to collect it from, and then you would fuse it with cancer cells. Now, because this question is all about the fusion of those two cells and has 3 marks, you would have to mention a few details, and that would just be cell membranes of the two cells fusing together by the help of a glycoprotein present in the cell membrane is known as PEG or polyethylene glycol, which facilitates the fusion of the two cells. To outline the use of monoclonal antibodies in the treatment of disease. Monoclonal antibodies, like any antibodies, are specific to particular antigens, so they have a shape, so they have antigen binding sites in the variable region that are complementary to the shape of the antigen that they want to work against. And so they are specific in that form. Radioactive substances can also be attached to monoclonal antibodies so that they can be used to detect the location of a particular cancer cell, for example. And then after binding with the particular antigens, antibodies do their job and stimulate killer T cells to kill the cancer cells. Linoleic acid is an unsaturated fatty acid that is found in some triglycerides and some phospholipids. Phospholipids are components of cell membranes. Figure 2.1 shows a molecule of linoleic acid, and part A is the composition of cell membranes of plant cells of plants changes in response to changes in temperature. That's because change in temperature changes the fluidity of membrane, and so to counter that effect, the composition of cell membranes changes. At the start of the cold season, there is an increase in the proportion of phospholipids with unsaturated fatty acids in the chickpea Caesar eritinum. Chickpea plants that do not make this change do not survive. So just how the increase in proportion of phospholipids with unsaturated fatty acids help plants such as chickpea survive decrease in temperature. So decrease in temperature once again decreases the fluidity of the membrane, and to counter that effect, the proportion of unsaturated fatty acids increases, and that prevents too close packaging of phospholipids and thereby increases the fluidity which is needed, because if the membrane fluidity decreases, that would damage cell. B1, state why triglycerides and phospholipids cannot be described as polymers, because polymers are larger molecules made up of repeated monomers, which phospholipids and triglycerides are not. 2, state two differences in the structures of triglycerides and phospholipids. So triglycerides have 3 fatty acids as opposed to 2 in phospholipids, and triglycerides also do not have a phosphate group unlike phospholipids. C. Platelets metabolize linoleic acid to produce a molecule known as thromboxane. Thromboxane is released by platelets when blood loss occurs. Thromboxane acts on smooth muscle cells in the walls of arteries. This causes arteries to constrict, which reduces blood flow. Explain why constriction of arteries following blood loss is an example of cell signaling. Alright, so thromboxane over here is the cell signaling molecule, since it is the molecule that causes the change in muscle cells in the first place. And to answer questions related to cell signaling, you need to outline the entire process and all of the components or the substances that are involved in it. So first things first, thromboxane over here is the cell signaling molecule that binds to receptors on the target, which are muscle cells over here, and then it elicits the response, which is contraction of these muscle cells, which in turn lead to constriction of arteries. Now keep in mind that thromboxane, or the cell signaling molecule, does not directly cause constriction of arteries. It, however, causes contraction of muscles. The response that it elicits is the contraction of muscles, which in turn causes constriction of arteries, which is controlled by contraction of muscles. 3. Nuclease is an enzyme that is used to hydrolyze proteins in solution. When the enzyme is mixed with a 2% protein solution, the reaction mixture changes from white to colorless. So that means in the presence of protein, it is white in color, and once the proteins are hydrolyzed to smaller polypeptides or amino acids, it changes color to colorless solution. A student carried out an experiment to find the effect of copper sulfate and potassium sulfate on the activity of the enzyme. The student made 4 reaction mixtures in test tubes A to D. Test tubes A to C were they contained equal volumes of protein solutions and 0.1 cm cube of solutions of copper sulfate or potassium sulfate. Test tube D contained the same amount of protein solution and 0.1 cm cube of water. So all of the test tubes contain the substrate or the protein for the enzyme nuclease. D contains only water, whereas A to C contain either of the 2 solutions of copper sulfate or potassium sulfate. 0.5 cm cube of a 1% in the enzyme solution was added to test tube A, which contains the substrate as well, and immediately placed into a calorimeter. The calorimeter was used to measure the intensity of light that is absorbed by the solution over hundreds of seconds. The procedure was repeated with the other reaction mixtures B, C, and D. You've got substrate in B, C, and D, and then you add the enzyme, which is going to change color from white to colorless when it hydrolyzes proteins into amino acids. And when that does happen, the color change causes difference in light absorbed. White color absorbs the greatest amount of light, and as it gradually converts to colorless solution in the presence of proteins as they are converted to amino acids, the light absorbed by the colorless solution is lower, and therefore the absorbance of light absorbed decreases with time as proteins are converted into amino acids and the solution changes color from white to colorless. As you can see in the graph, the greatest change in absorbance is in the test tube D, which contained only water. Test tube C differs only a bit, which contains potassium sulfate, and A and B containing copper sulfate solution have the greatest difference from the test tube D. A1 suggests and explain why the measuring why measuring the absorbance of the reaction mixture over 100 seconds is a suitable method for determining the activity of nuclease. In other words, you need to know why the span of 100 seconds is appropriate to observe the changes that represent the results. And that's because 100 seconds are enough to see the change in absorbance of light as proteins are converted into amino acids, which is also proven by the fact that the change in observance is shown in the graph over 100 seconds, and you can compare the results. 2. With reference to figure 3.1, describe the effects of copper sulfate solution and potassium sulfate solution on the activity of nuclease. Suggest explanations for the effects that you have described. So with time, the enzyme converts proteins into amino acids, and the absorbance decreases as the white color fades into a colorless solution. In solution A and B, due to presence of copper sulfate, the absorbance does not decrease as much as it does in solutions C and D that do not contain copper sulfate, meaning that copper sulfate decreases the activity of nuclease, and the solution with higher concentration of copper sulfate, or A, reduces the enzyme activity more than solution B, which has a lower concentration of copper sulfate. And potassium sulfate does not decrease the activity much, as you can see that there is not much of a difference in solutions C and D, where D does not contain anything other than water, substrate, and the enzyme. And as for the explanations, you know that a substance which affects or decreases an enzyme activity would be an enzyme inhibitor, and so copper sulfate over here, which causes decrease in enzyme activity, would be inhibitor of nuclease enzyme. So it could be either competitive or a non-competitive inhibitor, binding either through the active site or the allosteric site. B. Nuclease can be immobilized in alginate. Immobilized nuclease is used in the food industry to produce foods with high nutritional content. Explain the advantages of using immobilized enzymes such as nuclease compared with using the same enzymes free in solution. So immobilized enzymes can be reused since they are not lost in the product, and they do not contaminate the product since the product is kept away from them. 4. Meristemic tissue is found in the growing regions of plants such as shoot tips. Meristemic cells have a similar role to stem cells in animal cells. So meristemic cells, like stem cells, can divide to form cells that can differentiate into particular cells. Figure 4.1 shows some of the stages in the formation of a mature phloem sieve tube element and companion cells from a meristematic cell. So you've got all of these cells in figure 4.1. A cells E and F in figure 4.1 are daughter cells produced when the meristematic cell divides in the shoot tip. Explain why it is important that one of the daughter cells, or cell E in this case, is a meristematic cell. Once again, you're supposed to describe the importance of these cells, and we need meristematic cells so that they are able to divide and form cells that can differentiate into particular cells. Complete table 4.1 to describe the changes that are shown in figure 4.1 between the stages F and G, G and H, and H and J. Between F and G, so this is the meristematic cell, and this is the cell that is supposed to form the companion cell along with the phloem sieve tube element. Cell F elongates to form cell G, and F did not have any vacuole, which G does, so a vacuole forms between F and G. Following that, between G and H, the single cell divides to form two cells, one of which does not have a vacuole, which is going to form in the companion cell in the future. And between H and J, the nucleus of the maturing sieve tube element is lost, and the companion cell, or the maturing companion cell, divides to form two cells. Between H and J, the two cells also elongate and become much larger. C. Explain how the structure of a mature sieve tube element is related to its function. So the sieve tube element does not have any nucleus, vacuole, or really any large organelles, which reduces the resistance to flow of the phloem sap. The sieve plate pores in the sieve plate allow phloem sap to flow between sieve tube elements, and the intact cell surface membrane of the phloem sieve tube prevents loss of assimilates, since the cell membrane controls the movement of substances, and plasmodesmata between sieve tube element and companion cells allows loading and unloading of assimilates like sucrose. We describe the functions of companion cells in transport in the phloem. So companion cells have proton pumps that pump the hydrogen ions of inside their cytoplasm into their cell walls, and this creates a potential gradient, and that movement is caused by active transport requiring energy. Now that a gradient is created, the hydrogen ions would move back into the cytoplasm of companion cells down the potential gradient, and using a transport protein known as a co-transporter protein, which allows the movement of sucrose from spongy mesophil cells into companion cells so that they are able to diffuse from the through the plasmodesmata and into phloem sieve tubes. 5a. The mammalian circulatory system is described as a closed double circulation. Explain what is meant, why it is called closed and double. It's called closed because all of the blood remains within the blood vessels, and double because all of the blood passes twice through the heart in one complete circulation. B. Figure 5.1 shows a drawing of an external view of a mammalian heart. Two cross sections were made of the heart, where section 1 was made across the line AB over here and section 2 was made across the line CD. Drawings of the two sections were viewed from above as shown by the arrow on figure 5.1. Figure 5.2 is a drawing of a section AB and 5.3 is a drawing of section CD. One name structures 1, 2 and 3 shown in figure 5.2. So 1 over here is this vessel over here, since it's next to B, and since it is below the pulmonary artery over here, this vessel would be pulmonary vein. 2 is a semilunar valve, since those are the valves between ventricles and arteries, and 3 is the right atrium, since it's just above the vena cava over here, so it's present on the top right corner of the heart. To explain why the wall of chamber Y is thicker than the wall of chamber X as shown in figure 5.3, so chamber Y is going to be left ventricle if it's got a thicker wall, and also it's present on the left side of the heart, which is D. And that is so because the left side of the heart or the left ventricle has to pump blood around the body through the systemic route, which is longer than the pulmonary route and therefore has a greater resistance. And to overcome this resistance, the left ventricle has to exert greater pressure allowed by the greater force generated by the muscles in the left ventricles. C. Explain how the contractions of the chamber of the heart are coordinated during one cardiac cycle. So the sinoatrial node, which acts as the pacemaker, sends out an excitation wave that causes contraction of both of the atria at the same time together, and then the excitation wave has to pass down to the ventricles, and so that is done by atrioventricular node that relay the impulse after delaying it for a 0.1 second. So atrioventricular nodes relate to the Purkinje fibers that conduct impulse down to the base of the septum, then up the muscles in the walls of ventricles, so that both of the ventricles contract together from the base upwards. 6a. The DNA in the nucleus is known as nuclear DNA. 1. In the cells of the grasshopper Cortopus brunis, 20% of the nucleotides in nuclear DNA contain thymine. Calculate the percentage of nucleotides in the nuclear DNA of C. brunis that contain guanine and explain your answer in terms of the structure of DNA. So if the 20% of nucleotides are thymine, that means another 20% would be adenine. Sum that up and you get 40. Subtract that from 100 and you'd get 60. And half of those are going to be guanine, whereas the other half are going to be cytosine. So you have the percentage of guanine as 30, and the reason is due to complementary base pairing. Adenine binds with thymine, guanine binds with cytosine, because of which they have equal percentages. It binds with adenine binds with thymine, guanine binds with cytosine, because of which they have equal percentages. To state another location other than nucleus where DNA occurs in cells of Cybernus, so DNA is also found in mitochondria. B. Figure 6.1 is a diagram of a molecule of tRNA. The region labeled R shows detail of the part of the tRNA molecule. One complete figure 6.1 by writing the sequence of bases in the region labeled R. So C has the complementary base G. The complementary base of A in an RNA molecule is uracil. Guanine binds with cytosine. To state the name of the region Q and explain the role of region Q in translation. Region Q is the anticodon that binds with codon on the mRNA to bring the specific amino acids that are coded by a particular gene to form the right amino acid sequence in the polypeptide. 3. State the function of region P. So region P over here is the binding site of amino acid. Alright, this is it. We are done with this paper. Thank you for watching.